Thank you. Okay, so it's been a while since we met.
Yes.
And because I mean, Yuhang was super busy with finishing off and submitting thesis, and so I think that took priority over his very capable previous organization of meetings. So uh, but we things have been happening in the background. So what I was hoping we could do today is just have Yuhang talk a little bit about what he has been doing with summarizing the work on his main project
Mhm.
in the last few months and think about potential next steps. Then there was a bunch of new data from Warwick on the TB enzyme. Laura, if you wanted then to expand on that and talk about what you've been doing with the TB side of things, hopefully. And then Jarvis was going to talk to us a little bit about a potential project that he is running at Imperial and how that might fit in with or benefit from an involvement with open source antibiotics. All right, if that sounds okay, Yuhang, did you want to run through some slides and get people's views? That would be super.
Yeah, sure. 1 second. Two. Um, and you see the screen. Sorry. Uh, oh,
Yeah, great.
okay. There's some security reasons. So,
Yeah.
right. Nice to meet you guys. So it's been a while. I'm about to talk about my PhD work, which is optimizing the ligase inhibitor for retained potency, improved accumulation, and potentially multi-target purposes. Uh, our work started off with the mu ligase, which is uh,
Remember, Yuhang, it's like 20 minutes or so, right? So you can see,
okay, yeah, sure.
we all know it, right?
So basically, so basically, our project started off with with the mu ligase as the target because they have shared mechanism, conserved binding site and great selectivity because they're absolutely human. And more importantly, we have a promising music series to start off. this series were this series were paused because they were ineffective against the wild type bacteria, and their overall performance against the efflux deficient strains were also not ideal. So we are taking off from that. Uh, to start off this project, we first need to because this will be a structure-based drug design project, so we need to figure out the binding modes. Validate. We need to figure out the binding modes. The original paper focused on the homology model, using a homology model, but when we started our project, uh, we've already got the structural. This is the original paper with the homology model. But by the time we started off our project, SSG study already donated several crystal structures, and the binding mode they're quite consistent. The binding mode was actually a flipped version of the predicted binding modes in the original paper. So we sort of believed that the crystallography was more convincing, but the one thing that wasn't sure about at the time was because the C-terminus domain was truncated for crystallization purposes. So we weren't quite confident that whether this flexible domain being truncated would have some effects with the overall binding pose or not. It could potentially introduce some like a human artifacts in the overall generation of binding pose. So we utilized homology model by ourselves. We carefully modify the modify the homology model, and in our case, we did not overly minimize the semi-flexible structure, and eventually, eventually we managed to predict the binding poses, were further validated by the most recent crystal structures from our team. So the RMSD were pretty good. So showing that our homology model was actually better and more indicative. And based on this, we generated the 2D map for the E. coli and the Pseudomonas aeruginosa to guide our future structure-based drug design. And we can see two directions. One direction is the solvent direction, where we can grow our ligands into the solvent for more functionalities, such as improving the accumulation improving the accumulation performances, which we will mention later. And the other side is to grow into the phosphate binding site to target the conserved helix across multiple ligases, and this will serve as the anchor for multi-targeting purpose. Okay, so the first bit of our project is to utilize the existing predictive models for the accumulation, for improvement accumulation. So compounds in the AstraZeneca original work, we've identified two potential barriers that could be the cause for losing the wild type antibacterial activity. The first bit is the dual membrane structure of the gram-negative bacteria. The other part is the efflux. So we with the established Hergenrother model, which are very easy to install and easy to follow, we decided to take on these two rules to improve our uptake in order to achieve better antibacterial activities. However, these two rules were mainly focused on improving the outer membrane uptake. They did not cover the inner membrane penetration. That this is not guaranteed, and so far there still haven't been a universal rule or established at the same level as the Hergenrother rules to explore the inner membrane penetration. And from the original AstraZeneca work, we found that after applying sub-inhibitory colistin to permeate the outer membrane of the wild type E. coli, the MIC improved. No matter the compound was not having a charge or having a charge. So this gave us confidence that the bearing positive charge might probably still have a pathway to go through the to permeate through the inner membrane. Could probably be like some anti-dependent pathways which are utilizing, for example, proton multiforce. So at this stage, we made a speculative hypothesis that these structures probably won't matter. Probably won't have the pathways for the inner membrane, and we just need to focus on whether these structures are meeting the criteria for outer membrane penetration. And after examining with these two models, we found that there are several aspects of the physical chemical properties not quite matching up. Therefore, we decided to modify the whole series based on these two sets of rules to improve accumulation. with this idea in mind, we know that we are gonna install the overlapping feature of the two rules, which are unhindered positive charge. The best way to do the best position to install this was through this side chain, which we have is the solvent exposed direction, so that so this positive charge could be adopted by the solvent to avoid desolvation penalty. Yes, to save the Ki. And there are other aspects. There are other aspects of physical chemical properties being explored just to visualize the individual influences towards the accumulation. And the most direct bit of this modification was to change the alcohol straight away to modify the best hit on B by changing its hydroxyl group directly to the amine group and the guanidinium group to have the minimal structural changes for the best potential retention of the enzymatic potency, while it's hopefully improving the accumulation. So therefore, we've synthesized these compounds. I'll just skip over this, and we've managed to obtain a general trend that that this series were maintaining a general dual targeting SAR profile, but the best retained enzymatic the structures that retain most enzymatic potencies were the 2A and 3A. There's one outlier. There's one outlier unexpectedly hitting the E. coli MURC at a comparable level to the 3A, which is also comparable to the original alcohol binders as the controls. So, but these three are the ones that we focus more, because they review the most straightforward structure transformation, and they have been proven to retain the most enzymatic potency through this among the entire series. Our docking studies further revealed that the pocket, despite the pocket was conserved, but some key residue differences might contribute to the differences in the E. coli and the Pseudomonas MURC inhibitory potency. So we can see the corresponding arginine, threonine, aspartame, alanine. Those changes may promote conformational changes drastically, and this explains why this compound has like a strong preference only for the E. coli MURC. As for the other three structures, the change was not significant, but sort of influenced the overlap of the phenyl ring in the hydrophobic group area. The next bit is our accumulation tendency. We've also managed, because we incorporated both sets of rules to the same scaffold, we also managed to get a dual accumulation profile, SAR profile, and from And from the plot, you can see that the guanidiniums and primary amines improved accumulation drastically in comparison with the in comparison with the alcohols. There are some compromised amines which were either substituted or conjugated. Those were not quite good in accumulation as suggested by the original rules anyway. And these rules were quite predictive, as you can see from the success rate. But the problem for this, the problem for these rules are like they cannot guarantee cannot guarantee the cytoplasmic accumulation because the assay only gathers this the whole cell accumulation numbers. Therefore, like for compounds that can only penetrate through the outer membrane can be a potentially false positive. Okay. These are the accumulation data fitting with the rules, and there are some structural features are not matching up with the rules. We're gonna skip that. So after retaining certain level of Ki enzymatic potency and improved accumulation, what do we get? What do we get in terms of MIC? So we can see that in the alcohol subsets, the general tendency to in the alcohol subsets, the correlation, despite weak but still like noticeable, between the enzymatic potency and the MIC. So showing that like this subset might rely on the enzymatic potency to exert the antibacterial activity. But when we overlay the accumulation data with the MICs, we cannot see strong, clear correlation among the entire series, except for the guanidinium series. You can see a vague trend that's like that's having a higher accumulation could potentially improve the MIC. But that does not guarantee the target involved. That does not guarantee the involvement of the target was actually the MIR-C. We're gonna talk about that later. Here we can see these 6 highlighted seven highlighted compounds are the most useful, the most indicated ones. The intracellular accumulation was actually improved as we transform from the alcohol to the amine and guanidinium, but the enzymatic potency dropped. Therefore, but the enzymatic dropped, and in terms of the MICs, these barely showed any improvement, but only only a worsened value. So we are so for these direct transformations, the modification was not quite successful, but very surprisingly, we can see this 3F compound. It doesn't have like a strong. It doesn't. These 3E and 3F. They don't have that strong Ki values. But they only have like a good accumulation, but they have surprisingly strong. Well, these are actually the good ones among the entire modifications. So I can't say it's surprisingly strong, but totally unexpected to have at least this like a 3 to 4 folds just 3 to 4 folds different from the best hit in activity against the E. coli efflux deficient strain. So this may potentially indicate that these compounds have a secondary target, or yes, exerting the antibacterial activity via a murating independent route, or it could potentially, yeah.
Okay, you're gonna have to finish up, I'm afraid.
Yeah, sure, for sure. So that's all. These are our special cases. We've already covered. That's all. Right.
Okay, cool.
Okay, cool. Yeah.
So the idea is that obviously Yuhang is drafting up the paper, and we're going to send that around ASAP to identify any last data that we need. But yeah, I mean the accumulation data provided a real insight into the potential values. Any questions anyone has about that? Just straight off, or shall we continue?
Have to continue. My only question is, Yuhan, how much longer are you around for?
I don't know, forever.
Yeah. Submitted revised thesis is where he's up to.
Yeah,
I see.
so far. Yeah.
Okay.
All right. Thank you, Yuhang. Great. Paper is coming around very soon. All right, Adrian, would you be happy to walk us through the data that you sent around yesterday or the day before? I forget when it was.
Yeah, certainly. So
here we're interested in the corresponding activity against the TB enzyme.
okay, so well, I can share my screen.
After having had a sniff of activity against the pathogen by one of Yuhang's guanidinium compounds, which was compound number 88.
Right, so sorry, it's a bit slow here. Just give me a second. Yeah, froze.
Nothing yet.
No, sorry, you're all frozen. Hang on. I think I'm going to have to go back out and come back in again.
Do you want me to open the presentation for you, Adrian?
Actually, that might work better.
Yeah.
Okay, let me.
Okay. Right.
Okay, right, let me share. Fuck, it doesn't let me share it. They changed our
right. Give me a second. I've got a PDF version, which might be easier. Just 1 second. Right. So. Okay. So. Right. Can we all see that?
Hopefully, if you could, yes, yep.
Okay.
Yeah. Take it away.
All right. So we're going to talk through some of the work we've done with Mycobacterium tuberculosis, MERS-C, and Yuhan's compounds, and some work that we've done on some of the transition states analogs that we've been working with. So to start off with EUHANS compounds. If I just scroll down to the next slide. So I always actually put in the methodology that we use with all of the assays that we do. And for those people who don't know, basically the assays we run basically link the activity of the enzyme, which generates phosphate, to a coupling system which generates resirufin, which is a fluorophore. So they're all continuous fluorometric assays. They're all done in a 10 microliter volume on a plate reader. So working with, beg your pardon. So, okay, so working with the Mycobacterium tuberculosis enzyme, we characterized its response with a known inhibitor, which is an analog of ATP, which is ADPCP. So the top three panels are the results of that type of characterization. So we have on the left a dose response curve, an IC50, which is around about 4.1 micromolar. We plotted that out on a log scale so you can see the points more evenly distributed. And the graph on the right-hand side is the conversion of that data to a form where you can calculate a Ki, which it turns out to be about 3.1 micromolar. Whenever we've done this with a Pseudomonas enzyme, in a second, we have slightly different data in that the sense that it does not fit a single binding constant. So there's more than one IC50 in response to ADPCP, and there's more than one Ki. And we have seen this not just enzymatically, but Laura, I think, has also recorded it in SPR measurements of ATP binding. Basically, it almost implies that there's more than one type of binding site for ATP on this enzyme. The origin of that is something that we're trying to tie down. So looking at EUHANS compounds now. So we were tasked to try WYH88. We also did 87 and 85. So WYH88 was the only compound that actually gave us any significant inhibition, with an IC50 of 50, just over half millimolar, and a Ki of just under half millimolar. And that compares with values of IC50 of about 23 micromolar and a Ki of 17 micromolar for the Pseudomonas enzyme. When we tried other compounds in this series, for example, WYH87, which is an isomer, I think, of 88, we lost any inhibitory potency against the TB enzyme at all, which compares to the robust inhibition we're seeing for the Pseudomonas MRC. And you can pretty much say the same thing for compound WYH85. So I believe it was compound YH88 that gave an impact on MIC with Mycobacterium smegmatis. The in vitro data I've just presented will kind of suggest that it's unlikely that that MIC was brought about by inhibition of MRC. That's speculation consistent with the data, but it certainly isn't a potent inhibitor of the TB enzyme.
Yeah.
So at Warwick, we've also been interested in looking at transition states. We've been interested in looking at some of the old work that was done with phosphonates and the MER ligases, and seeing whether we can push that any further in any translational sense. So I'm going to skip a slide here because we're probably going to run out of time. But essentially, we're interested in looking at the crystallography of these molecules. with MER ligases, MER-C and MER-D, for example. And we're also interested in looking at the transition states of both parts of the MER ligase reaction, and also the intermediate phosphoryl MER compounds that lie between phosphorylation by ATP and the transfer of the group from the phosphonate to the amino acid. So if I just scroll down a bit. So there's good literature present as well for this type of work. So there's another ligase called gamma-glutamyl cysteine synthetase, and essentially it carries out a very similar sequence of reactions, formation of an acyl phosphate by transfer of phosphate from ATP to generate a first transition state, which is this pentavalent arrangement of atoms here, and then a second transition state, which is the attack of the amino acid on the acyl phosphate to form this tetrahedral center, which collapses to a planar one. And you can draw the same sort of analogous structures for the MER ligases on paper. Anyway, and this is with MER-C, basically the formation of a pentavalent phosphoryl intermediate, which you could potentially mimic, for example, with a metal trifluoride, and then the transfer of the amino acid onto that acyl phosphate with the displacement of phosphates, and you get a second transition state. Again, a tetrahedral center collapsing to a planar one. So what we're talking about basically is mimicking of this tetrahedral transition center in the second transition state with a phosphonate. And this work has previously been published for MER-C and MER-D and MER-E, although we haven't considered MER-E in this body of work. But basically, we sought and got a contract synthesis of the two compounds on the right-hand side by a CRO, Synthesis MedChem. So we've synthesized the transition state for synthesized the transition state for MER-D, which basically is the UDP muramyl alanine. Then there's a phosphonate here, the tetrahedral transition state mimetic, and then the amino acid. And this has generated a spectacularly low IC50 of about 17 nanomolar with a computed Ki of around about 1.4 nanomolar. So these are some of the most potent compounds we've actually dealt with thus far. We've then gone on, he said, skipping too quickly, to look at, sorry, beg your pardon, the MER-C transition state between UDP Merlac and alanine, and there's the phosphonate there. And again, we get nanomolar IC50s and nanomolar Ki's. The actual values of these constants are not altered at all by increasing concentrations of the amino acid. And in the next slide, they're not altered at all by increasing concentrations of ATP. So those are the Pseudomonas enzymes, and the Pseudomonas aeruginosa uses an N-acetyl muramic acid-based peptide glycan. TB, however, uses a peptide glycan that's partially N-glycolated, and the difference basically is the addition of a hydroxyl group onto the acetyl group of the miramyl sugar ring. The structures here are the two precursors we're talking about that will ultimately form an N-acetylated peptide glycan and an N-glycolated peptide glycan. We've done the kinetics of TB-MRC with both substrates, and the N-glycol group and that hydroxylation causes a massive drop in the Km from 1.5 millimolar to 43 micromolar, with little loss in actual turnover, but the efficiency of the enzyme basically drops significantly, going from a glycolylated substrate to a burnback substrate. So essentially, if you compare, for example, the TB enzyme trying to be inhibited by the MRC transition state we've just talked about, which is a MRNAP-based molecule, the TB enzyme is far less sensitive than the Pseudomonas enzyme to that particular transition state mimetic. However, we also had made the TB transition state with for MRC together with the N-glycol intact. And what we do see, again, it's strongly inhibitory, and we do see a preference for the N-glycolation. So the work is currently ongoing, and Laura's going to tell you a bit more about some of the structural stuff. The one last thing I was going to say was, way back when, one of the things that started off phosphonate or interest in phosphonates in terms of amino acid ligases was the seminal work on DDL, where basically this phosphonate as a mimetic for a transition state for the formation of dialanine alanine was synthesized, and in the presence of DDL, it crystallized, and in the presence of ATP, this phosphonate was itself phosphorylated. We've tried to mimic this with MRC and MOD, and at the moment that's challenging, partly because this has to be done with stoichiometric quantities of enzyme, and we have issues with background rates at the moment in enzyme assays at those enzyme concentrations. So hopefully we can resolve this ultimately crystallographically. Talking of which, I'm now going to hand over to Laura. So if I may stop sharing.
Thank you. Can you see the screen?
Yeah.
Yeah.
Okay, good. Right, so I'm going to give you a little update of everything we've been doing for the past few months since we last talked. So I'm going to start with the transition state structures that we got for Pseudomonas, MRC, and MRD, and then the structures that we have gotten for the open source project, and then some stuff about tuberculosis, MRC, and some other tangent analysis that we've been doing based on what we have seen with these structures, the WYH. Right, so let's start with the transition state. In this one, we have saw structures for the Pseudomonas MRC at 1.9 nanometer resolution, which is quite good. And we can see a lot of nice interactions and some waters that might be involved in either binding on catalytic, especially here in these regions. I'm not going to go deep into all the details because we need to go through all the slides and then continue with the meeting. But yeah, these proteins, the Pseudomonas MRC, they're forming dimers. We can see this dimer formation as well in solution with different experiments. We also saw the magnesium binding on the structure, so everything was really good about these models. And then we saw that this dimer looks very similar to the apoprotein form of E. coli MRC, which will come relevant later. Then in the case of the Pseudomonas MRD structures, these ones are monomers. And for this one, we've got two structures, one with the transition state only, at a little bit better resolution, 1.7, and then the other one was 2.6, and we also had an ADP molecule in it. We had a second ADP molecule in a different pocket that is created with another Siemens-related molecule in the crystal, but I'm not sure if that's relevant in terms of biology. I think it's more related to crystallization bias, an artifact, right? So we've got this structure as well, and in this case, we've got many more waters to look at, interacting, like water interacting together with the protein regions and also with the ligand. So those could be some catalytic waters that we could look into as well. In one of the structures, we also found some potassium in there, but this one is coming from the crystallization condition, so I don't think there is anything physiological about this position here. It's just a nice pocket where things can bind. Right, and then let's move on into the next step, into the next part of the talks. So I just want to remember, this is what happens when the ATP binds. So you've got an apo structure. I'm trying to make it start again, but well, you've got an apo structure which is open, and then when the ATP binds, it closes on itself and making a more compressed conformation. This is the structure from influenza, because it has a really nice description on the MERSI structures in the literature, and also in terms of how many structures there are. So that's why we took it as to show you what happens, right? So when we look at the structures that we got for MERSI, let's start with the E. coli MERSI with your inhibitor with 30-1P. First of all, this structure was quite difficult to make. As you can see, there are 6 different molecules. That's the asymmetric unit, the actual model that we need to work with. Gabriel got this structure, and he had to build a lot of these chains because this was a new conformation of MERSI. This is 6 subunits that are forming 3 dimers. So E. coli MERSI is also a dimer in this case, okay? And the solution is a little bit worse. Now we're going into the worst resolution area. Right, now we see this happening, right? In the presence of the compound, we can see the dimers actually moving in like close to 90 degrees. It's a lot of change in the case of the E. coli MERSI. And we can also see in here how the structure doesn't close, it opens. So we've got a more open conformation upon binding of the inhibitors. The dimer rotates as well, because this section here, which is the N-terminal of the protein, moves a lot, and it will clash with the normal conformation of the dimer. So this one has to be moving as well. And then we also observe that the binding site for the magnesium is mainly lost. Because this is all open, so opened, then the interactions that you have between the residues is mainly gone. The distances are really large. So yeah, we're thinking that maybe there's a new inhibition mechanism that we haven't seen before, and it might be involving the loss of the binding to magnesium. We'll see, we need to do more analysis. Right. Inside that structure of 6 molecules, we actually can see different conformations of MERSI, E. coli MERSI. And then we will leave one of the chains, which we called F, is the one that is the final binding conformation. And the other ones are just transition states from the APO to the final bound conformation. This is because of how the B factors behave, both in terms of the ligand and the protein. The B factors just tells you how much the atoms are moving in that location. So the smaller the B factors are, the more rigid those molecules are in those positions. Right. Now, we also got recently, very recently, the structure for the 33G. We haven't finished the refinement. We haven't even started the refinement because, as you saw, there's 6 structures, 6 molecules to look into, and the resolution is a 3.4, which doesn't make things easier in order to refine. Also, we see a blob, but it's only for the core part of the molecule. We don't see the whole molecule. We are going to refine this, and we hope to see more of the molecule the more we refine the structure, because there are some sections that need to be rebuilt again, because, again, we've got conformational changes related to this open state. So we will share this once we have it, but I don't think we're going to see the whole molecule for this 32D structure. Now, we also got, these ones you are aware of, we also got the Pseudomonas mercy structure with the compounds 78 and 77. In this case, we're going to compare with the structure that we already have, which is with the transition state. The structure of the Pseudomonas mercy and the transition state, as I mentioned before, looks like the APO structure, right? So, as you can see, again, we've got an opening of the structure and then also a shift in the dimer position. So this is consistent within different organisms and different molecules that we've got for the series of these WYH compounds. So it could be a different a mechanism of inhibition. We want to try this with other inhibitors, but it's very difficult to do this with inhibitors that are not really good. So we need good binding for the compounds to be able to do this analysis. But we're looking into it. We're going to also do some analysis with the ADPCP, which is an inhibitor of the reaction, and then see what happens when we've got the ADPCP and what is this change in the conformation. We can't get the structures for Pseudomonas, ADPCP, or E. coli. We don't know why, but we can't get them. But we did have some cryo-EM data for the Pseudomonas mercy and ADPCP. I'm in the process to be able, because I moved to Spain and the data is in the UK, so I'm in the process to sign some documents so I can actually analyze the data in the UK and then do this analysis, because the data is good enough that I would be able to see if at least the dimers are shifting or not. Now, we've been doing a lot of work on TB as well, on tuberculosis, MER-C, MER-D, MER-E, and MER-F. Okay. In the case of MER-C, we saw the structure as well, again, resolutions of 3.2 angstroms in the presence of ADPCP. We saw the ligand bound. The resolution is not great again, so we want to get it better. Now, this was one of the first crystals that we got, so we were optimizing the crystals at the moment. We don't see any conformational changes between these tuberculosis proteins and the APO form that comes from the bovis structures, right? Mycobacterium bovis and Mycobacterium tuberculosis have exactly the same sequence, so we got the same results really when we did the structure as expected. These structures, these conformations are similar to the APO E. coli MER-C. We don't know if this lack of conformational change is due to the protein itself and how it behaves, or if it is actually a crystallization bias. We're following this up with some NMR, and we already did the first experiment, which looks promising. So we will follow that up later. We're not going to try to solve the structure with NMR. We're just going to see what moves when we bind the ligands. Right, and then we're not very convinced as well about the ligand conformation. We think we've got some flopping around there, but yeah, we need better data for this. We're not going to say anything more about that because we need more data and better data. We did get 2-point-something angstrom resolution crystal, but it got destroyed at the synchrotron a few weeks ago. There was a problem with the synchrotron, and we lost about 5 crystals from different projects, like the other labs as well. And one of them was this nice crystal that had ADPCP and the N-glycyl substrate for TB MER-C. So it's a pity. We're going to try to do it again, and we will probably be sending crystals to Diamond because we don't trust the synchrotron here anymore. But yeah, then we also did some quaternary structure analysis because we were a bit confused about why the MER-C TB was showing as a monomer. So we did a little bit more of analysis with analytical size chromatography. These are the results. I'm not going to go into all the details of the experiments. But as you can see, we've got here Pseudomonas and E. coli showing as dimers. And then here we've got the TB MER-C, which is a monomer. Right. So now we've been doing some more analysis in terms of which MER-Cs are dimers and monomers. So we've got here some proteins that we think or we believe they're dimers. and they are structurally validated and validated and also experimentally validated. Like, for example, Pseudomonas and E. coli, we do have the transcription chromatography profiles, and we are sure that these are dimers. And we also have some monomeric MRC structures that we checked. These would be the Mycobacterium ones. The Streptococcus pneumoniae, there isn't a structure, but we have already purified the protein, done some experiments, and we think it's a monomer. We also got some crystals. We hope they are useful so we can solve the structure. Then Dermatoga maritima is also a monomer as well, according to the structure. So we've been doing some analysis. So Gabriel has been doing this alignment of these structures that we already know the results, right? The dimers will be the red and the monomers will be the green. So we can see overall a great conformational, sequence conservation within dimers, especially in these marks are the residues that seem to be involved in the binding interaction for the dimers. So in these regions, we can see a greater conservation within the dimers and not really much conservation on the monomers, especially some of the regions like here. Right? So we looked into this a little bit closer. Again, we don't have a lot of time today, so I'm going to skip this one. Right, and then these are we identify some residues that seem to be very conserved within dimeric MRCs, and they seem to be conserved in the different organisms. So it would be on E. coli. We identify these ones, is the ones in orange. Then these ones in red as well. Is always the same residues, and the same for influenza, right? And if we overlap all the structures, we can see there is a great conservation, especially in this loop here. Now we've been doing like a quick analysis with other sequences, other organisms that we don't really know if they are dimers or not. So according to the sequence similarities, we've got some other extra dimers that seem to be dimers because of the conservation, right? The key residues are conserved within those organisms, and then others that don't really have much conservation in those regions, and we predict they are monomers. And if we look at how they are in a phylogenetic tree, how they will be correlating with each other, then we've got most of the protobacteria, they are all dimers, and then the rest are monomers. So there might be something there to look into. We don't know yet, but this is a very preliminary result. We will have to redo all this properly. So what next? We would love to get better structures for the WYH compounds, but I think we have to pause at this moment and reassess the stuff, because for the past 6 months since Gabriel started, we tested more than 200 crystals for them. Before that, I've been testing crystals. So I think we tested a lot of stuff. The resolution is not great. We can get crystals, we just cannot get the resolution good enough. So we are stuck with E. coli most of the times at 3 angstroms. Yeah. At 8 angstroms. Then our question is for crystallography, do you think we have everything we need for the paper? Should we also split the dimer thing for a second paper? And then maybe talk more about the mechanism of the inhibition there. I don't know. We'll have to discuss probably after Johan has the paper drafted. It will probably be more clear when that's done. And then in terms of the TB MRC, we are optimizing, trying to get those nice crystals back again. We are also preparing new constructs for the expression to get different tags and try to crystallize with different tagging situations. And maybe that will make the crystallization a little bit easier for this MRC, because we've got a lot of plans for structures of TB MRC, so we would like not to struggle as much with this protein. And we did get some UDP and glycoaluronic acid crystals, but I will need a little bit more optimization, and hopefully next time the crystals won't be destroyed as in front. But yeah, that's everything.
Yeah, it's a lot. Thank you. Wow.
Yeah.
Busy. For the paper, yeah, the draft will come around. That will clarify what's missing. I think the dimeric analysis most likely belongs in a separate thing, but let's see what you
I
think
think so
in the
too.
draft. Yeah.
Mhm.
Any other quick questions? I'm conscious that I'm very keen that Yav has a chance to talk, but
Yeah, I'm happy to stay.
yeah, me too.
Yeah.
Imperial team, are you happy to stay beyond the hour? Just okay. Okay. Any other queries to Laura before we switch? Thank you for all that. Okay. Then Yav, do you want to say who you are and where this comes
I
from?
guess let's try not to take too much of everyone's time. So yes, I'm Jarvis Smallfrost. I'm at Imperial in chemistry. But 7 years ago, I was working for a machine learning small molecule drug discovery startup company called GTN.ai. And actually, one of the things I was doing there, which might be of interest to Adrian, was doing rational design of transition state analogs with different methods. And I'm really a sort of, I guess, a theoretical chemist by training, and over the last 2 years we've been doing an increasing amount of sort of antibiotic design, starting with master students and looking at peptides because they're quite easy to synthesize, and building our own machine learning models and so on. And then we have Eric Kahn, who's here on an exchange program between Imperial and Hong Kong University, if I remember it correctly. And so he has some slides which he will show, sort of using these machine learning co-crystallization models through Bolt's Gen mainly to try and predict binders for ligase, and then also doing some Vena-based docking. And then there will also be a master's student in the next academic year who will be looking for small molecule binders for ligase with different methods, probably starting with docking and so on. And I'll also finally have a PhD student called Delena who's starting in September, and she'll be sort of just running all of our antibiotic work, so sort of coordinating, mainly doing peptide synthesis herself probably, but also sort of coordinating any small molecules and so on that we get done. So I guess the practical sort of strategic thing rather than just the science thing is that, Eric will hopefully by the end of his project in like a month or so have a load of peptides that we think are inhibitors, and we should be able to synthesize those starting in September. And then what do we do next? Would you like them? Who do we send them to? And all those sort of questions. Then hopefully over the next year, we might also have some small molecules, possibly even some transition state analogs. So I guess, that's just all the sort of high level strategic stuff. And then I see if I can share. Ooh, hopefully you are happy with this. I think it's this one. And then if we slideshow that. Is that visible to everyone? It looks like a slideshow.
Yep.
Okay. So
Eric. So I'll do the presentation, and I'm Eric, and I'm very happy to join the meeting here today. And so what I've done during the past month is to design peptides to fit into the Hin-Mercy UMA pocket, right, as suggested by Yu Hang actually. And so essentially the flow is to identify the pocket, which I defined it as 5 angstrom distance to where UMA actually sits in. And we feed the residues into Bolt's Gen design, and it generated 100 peptides, and I filter it according to the occupancy of the pocket. So I actually wanted to sit in the pocket, obviously, and we also did a scoring through Autodoc Vena to see the binding affinities of different peptides generated, and we had a sort of a ranking thing and of 10 candidates. So essentially we chose this UMA substrate pocket because it's large and it's very balanced, as we can see from the pocket chemistry. It's like 4 acidic amino acids, 4 basic, some aromatic, some hydrophobic, comparing to what I did at first. What I started with MERD, it's very, it's sort of cracked and very imbalanced, which resulted in a very imbalanced result. So we actually worked on MERC instead. And the figure on the right with the gray parts being the MERC, the in-MERC, and actually the pocket, and the blue one being the actual UMA substrate, while the orange one being one of the docked results. So as we can see, it actually inhibits or takes up the space of the UMA pretty well in that sense. And so essentially, Bolt's Gen provided from the 100 generations of results, right? There's the mean occupancy is 26%. It's not good, but we don't have to focus too much on that, because all we want is something up on the right hand side of 50%, I would say. So the distribution actually matters a bit, right? There's like about half of the designs with above 30% occupancies, about 16% of the designs above 50% occupancy, with the best one being 72%, which is not too bad in my opinion. And where the composition, we can also have a look, it's pretty balanced, right? As shown on the slides, right? 7.4% acidic versus 7.3% basic. No designs exceeds 40% acidic, in contrast to what I did actually before to one of the MER-D analogs. It's very cracked. It's like, I think it's like averaging of 30-ish% acidic versus 1.6% basic, which is not too good. And so here comes the 10 candidates, with the UMA one being negative 9.16 kilocal per mole, as illustrated by this dark green line, whatever, dotted line. And so as we can see, very interestingly, the best pocket occupancy peptide actually showed the best binding affinity out of all of the 10, which is, well, a thumbs up, I would say, for the results, given that models often go wrong, and this is sort of a thumbs up for the results. And this is the graphics of the leading peptide that we have. It's the TTDP GFGT thing. As the figure on the left hand side shows the best posts of it. The one on the right hand side shows the 9 return posts in total. And very interestingly, it actually, the 9 posts actually are not very close in terms of space, but they are very relatively similar in terms of energy. So I would say it's not a very accurate in terms of where it actually sits, but it's more like an energy-based tool. And with that said, there's also something worth noting or worth flagging, is that so I essentially did redocking control against UMA and the HIN-MERC itself, and it showed it actually lands about 5 angstrom away from the crystallographic position, the known ones. So it's a little bit off, but so I would say, with that said, I would say we'd like to treat the data more like a tier-based rather than a scoring function. So it's like some of them are in the top tier, rather than this specific peptide actually shows negative 8.34 kilocal per mole affinity in that sense. And we also did repeated measurements to the leading option, the leading peptide, and it showed a spread of 0.91 kilocal per mole, which is, in my opinion, also acceptable. And we also brought some questions, because we couldn't, I couldn't really separate the selectivity between MERC or MERD with my design. Like it doesn't separate really well. So I was wondering if it actually matters. And I also discovered that the longer the peptide generated, it actually fits the less into the pocket. So I was wondering if it's a model issue, or if it's just normal. And I would also like to ask if there's anything sort of known substrate that binds to the HIN-MERC really well, so I can sort of use it as a control and stuff. Yeah, and that will be all what I bring today. Thank you. Yeah, that's it.
All right. Thank you. So I mean, obviously these haven't yet had any experimental testing, right?
No, I haven't made them, or even thought of them yet.
Right. Maybe the Warwick team could comment about positive control for MERC in that site.
What well, I mean, besides ADPCP or some of the WYH compounds?
Yeah, those would certainly be fine for docking with Vina because all you need there is sort of the atomic structure. But I mean, Eric, were you sort of asking for a peptide that was already which I guess you wouldn't necessarily know or care about?
Hi, Joe. It's Chris. We don't yet have a peptide for MUC, but we're just embarking on a project with Bicycle Therapeutics for a bicyclic peptide. So we're screening CNA with them.
Yeah.
So that might happen. I was also thinking about buying the NEB phage display library and running that just coincidentally. That could prove helpful.
That'd be
so that's a possibility as well. But no, we don't have one sitting around at the moment.
Maybe I can add that. So Roger Levesque in the early 2000s did some peptide display work with the ligases and got some peptide inhibitors. And Colin Fishwick at Leeds developed a cyclic inhibitor, peptide inhibitor of MUD as well. That they may be worth looking into. They should be easy to find on PubMed, but if you have a problem, let me know and I can do the references.
Good recall, Adrian. I think Roger Levesque's came from a sawfly genome. Was that right? I can't remember. Was it a sawfly origin? I can't remember. Some insect peptide. I don't remember.
Got a question regarding all the data analysis. What structure are you using? Are you using the open conformation or the closed conformation of the structures?
You mean for the HinMerC one?
For the docking, yeah.
For the docking, I think it's a closed.
Okay, because I was having issues with these AI-generated structures for the ligases because it's very biased towards the open conformation. So if you're using the open conformation, you might not be seeing the proper pockets for it. So maybe it's worth checking.
Right. Yeah, but I think the docking with Autodoc Vina would have used just the actual crystal structure.
Yeah.
So
Okay.
I
think the protein mobility is the Achilles heel to all of the studies I think have gone on over the past decades.
Yeah, it's the bottleneck of everything.
Okay, very good. Thank you.
I was just interested though, just on the data where you had that peptide. It had so many different poses. I was just a little interested / worried as to how specific that really was, and if you actually took that peptide and ran it as in a negative control through the other ligases, what scores you would get with that peptide, for example, on other UDP bind site.
Which ligases did you say?
Any that have got the UDP binding site.
I mean, I think yes. So from the ones that Eric has tested, it seems that these things will sort of bind to any ligase we throw at them.
Yes. I don't know why, but
which is not, I guess, a great sign. Just have got sticky peptides effectively. And
another thing we'd like to ask is there any enzymes, particularly apart from ligase series, that's worth doing a control experiment on? Yes.
Well,
LIMU, it's a UTP utilizing enzyme.
Well, it depends what you want to go after, but we have one of the questions was, is it good to target multiple ligases? Answer, absolutely categorically, and almost don't bother if you don't kind of answer, because all of the current monotherapeutic antibiotics all multi-target. The ones that don't, like rifampicin, you get ridiculously high levels of frequency of mutations resistance. So you have to multi-target. However, saying that, if you have a big peptide with many binding sites, maybe you can overcome that. But vulnerability to resistance is the number one question before really starting any discovery program. How are you mitigating a single amino acid change in a target and just destroying 10 years of work? Just go talk to people at GSK with the Anacor boron compound for a non-essential editing domain in a tRNA synthetase.
Just back to your question about the sort of flexibility of it. Yes, it is quite concerning, but also, I mean, peptides are and should be quite flexible, and just from a sort of a phys-chem point of view, that flexibility will actually give them an entropic contribution to lower the energy in that pocket. Otherwise you've got a cost of immobilizing the natural flexibility of the peptide in solution if you've got it into a very stiff pocket that's not able to move in. But indeed, most likely what we're seeing here probably is just that the algorithm doesn't really know where it wants to sit, and it's not particularly tight binding. So.
In terms of controls for other, try other proteins and non-mu ligases, do you want controls like negative controls, or I'm guessing yes? Right.
I mean, certainly I know almost nothing about the biology, and so we don't know of any, and we haven't tried any, I guess is the answer.
I would just maybe try to get any, you could do a search on the structure and then see similar structure with that folding, like not on the ATP side, because you're going to find some kinases, DDL. I think GDL will also come up as well with some kind of similarity in the structure. But maybe in that section of the structure, you can try to find something that is some kind of similar. Anything binding to the U and P motifs.
Right. Yeah.
Things with big pockets. I think some the Nudix hydrolases, those ones might be a good family to look into as well. NUDIX. There's some hydrolases that they bind nucleotides, different versions of the nucleotides. It's like 24 proteins, and like half of them bind different nucleotides. So maybe that one, and those had a really large pocket as well.
I mean, ultimately, the question with peptides is therapeutic utility and stability, cost of goods, all of that good stuff. Which was we discuss and wrestle with Bicycle.
Chris, if we get anything from those Bicycle experiments, can we share it in this open source environment?
That would need to be a discussion with Bicycle. Yeah, but I would hope we might be able to.
Oh, absolutely.
Yeah, that would be great.
Just in terms of practicalities of any assays, if they are to be done, like, are there any constraints in terms of the phys-chem properties or anything? I mean, relatively low concentrations just in, I can't remember the solvent that Adrian was using.
I mean, the general experience suggests that most of these assays can tolerate something like up to 2-3% DMSO.
Okay.
So it all comes down really to the solubility of the compound in question.
Yeah.
What they're like in water, what they're like in DMSO, and what concentration of organic solvents we can get away with to keep a homogeneous solution.
And for crystals is similar. The least amount of DMSO we can use, the better.
Yeah, I mean, likewise. I mean, everyone loves things in water if they can get them.
Yeah.
Yeah. Especially mu ligases don't seem to like DMSO for crystallization. So,
yeah. Probably on another conversation, Jarvis, it'd be good to follow up conversations about transition states, synthesis and modeling. Because we've got things that are potent but definitely not therapeutic, and just they're not biologically tractable because of those phosphates. So we need we need alternatives. So be very keen to discuss how we might go about that.
Yeah.
The
Certainly.
studies are not in the PDB, but we could discuss sharing them.
Mhm.
Okay.
Thank you very much.
Fantastic. And you know, as things progress the next few months, as you said, the other projects are starting, then very good to stay in touch about all of that. Sounds great. Were there any other questions that people suddenly realized they had from previous talks that they wanted to ask before we depart?
Are we missing any data for microbiology for the paper?
Probably, and that's the real reason why we want to distribute the first draft, is to get that sorted.
Yeah.
So it's coming.
Good.
Yeah, I've posted the potential request under the GitHub issue. So basically, I think we probably missing the MICs for certain key, for example, the aiming and the guanidinium version of the AZ595 against Pseudomonas and E. coli wild type, plus the PA beta N. I think we want to see whether these were infected by efflux or just the inner memory penetration barrier. I think we probably want to single out the factors so we can make a conclusion instead of like suggesting these two other potential underlying factors.
Johan, if you send me an email, copy Adrian in, and then we'll talk to AMR screening team about going to do that.
Yeah, sure, thanks. I think I will wait until the first draft has already been sent over to Matt, and we're gonna discuss about it and collectively with all the requests, and then we send an email together. Is that okay?
Yeah, sure.
Yeah, thank you so much.
Okay, fantastic. Thanks for coming along. Great to see everybody, and we'll talk again soon.
Matt, do you have 2 or 3 minutes just at the end?
I do.
Great.
All right, see you, everybody.
Bye for now. Thank you. Bye bye, everyone.
Bye.